This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 308, recorded April 4, 2024. Wow, what a nice little rhyme we got there. April 4, 2024. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michelle Swanson.
Hello, delighted to be here, and welcome, Mark.
From Charleston, South Carolina, Michael Schmidt.
Well, hello, everyone.
From St. Louis, Missouri, Petra Levin.
Hello, it's great to be here with you all.
And returning from the University of Puget Sound, Mark O. Martin. Welcome back, Mark.
Thank you. Once again, TWiM-adjacent.
Well, when you were last on TWiM, did you already have started Matters Microbial at that point?
No.
Oh, so this is your first post-Matters Microbial TWiM.
It is.
So perhaps you'll take a minute to tell us about Matters Microbial.
Yes, please.
Oh, I would love to. It has always been a dream of mine to be a microbial voice. I will not say the microbial voice, but a microbial voice. And Microbe TV and Dr. Vincent decided that it would be – that was for you. That it would be
Professor Vincent.
Professor Vincent. I'm so sorry, Professor Vincent. I just get Doc Martin. So there you go. Anyway, so what I was going to say is I was given an opportunity to kind of create a podcast, and my focus is generally on one particular area that a microbiologist has been studying. I've had some former students of mine. I've had some very impressive speakers. I've learned a lot. I've had a wonderful, wonderful time. For me, it's recreational. I hope other people learn from the experience as well.
Yeah, it's doing well. I appreciate it very much. In fact, I was on it, so it was a lot of fun to do that with you.
Oh, yeah.
You should get all the hosts of TWiM on it as well, Doctor.
I have asked a couple of them.
And they have said no?
They didn't say that. They said they were busy, but I would love to have all three of you individually on this show. I'm booked up until June.
Good for you, Mark.
It's a good thing.
Great.
Yeah.
By the way, Doc Martin is great because there's a TV character, right?
There is, and he's very curmudgeonly, which I am not, at least in my public persona. My private might be a little bit different. And then there's also, of course, the shoes. That's why I have 8 pairs of Doc Martin boots.
Seriously.
I keep hoping for someone to make me the glow-in-the-dark bacteria on the Doc Martens. That would be perfect. My wife says I have a problem. I own more shoes than she does. But
that is a problem.
Well,
I agree with Jenny.
It's your only fault, Mark. It's your only fault.
If only.
Isn't that lovely? What a
And you're
lovely
always
thought.
ready to go to a factory floor because you have all the steel-toed boots you could possibly need. So
Yes.
I think this is a great idea. Since you are Doc Martin, you should have a unique kind of Doc Martin shoe just for you.
I think so. People
Are they all high boots, or they're different?
I wear what's called the 1460 style, which is the original style that they were English bobbies wore them a lot when they were first invented. They have all kinds of other styles, but I prefer those. I don't care for the knee highs. No.
Ooh, those would be tough.
Knee highs? No, that's too much. Yeah.
Yeah, I agree. If you're a pirate, it's a great idea, but I don't think otherwise. But yeah, I have tried to find out if I could hire someone to create a custom pair, but I fear that's probably cost prohibitive.
I saw somebody actually tattooing their Doc Martens, essentially using a tattoo gun to decorate them.
That's an interesting thought.
I don't think you could do it with, black ones. I think it would be very difficult, but you could do it with lighter colored ones.
Yeah.
Or glow in the dark ink.
We could crowdsource the funding.
Well, Mark, when I see you at ASM Microbe, I want to see what Doc Martens you're wearing. Okay.
I do. Yes.
All right.
And we should point out we are not sponsored by Doc Martin, the shoe company.
No, we are not. We are supported by ASM but we are not sponsored by any other commercial entity because ASM is a nonprofit entity. And nonprofits can support each other. So Microbe TV is a nonprofit, and we can pat each other on the back, so to speak. All right. We do have science for you today.
Oh, yes.
And that's one reason Mark is here. But before Mark, we have Michael.
And I'm going to start us off with something that is rather exciting for those of us who worry about familial cancers and colorectal cancer. And once you reach the age of 45, that delightful procedure called a colonoscopy. All of this has to do with the topic of today's paper, which is entitled A Distinct Fusobacterium nucleatum clade Dominates the Colorectal Cancer Niche. And this is one of the few papers that's in Nature that is actually open access. So you all can read along with us. The authors, who are Zepeda Rivera, Minot, Bozek, Wu, Blanco, Miguez, Mungi, Jones, LaCourse, Wu, McMahon, Park, Lim, Kempczynski, Willis, Cotton, Yost, Sinsika, Kycook, Dilworst, Sagata, Bullman, and Johnson. And they're at a number of institutions that will run us out of the SIPNet if I name them all. But principally, they're at the Fred Hutch and other fine institutions. And as you might surmise from our story today is going to concern this 1 gram-negative rod. It's a strict anaerobe, Fusobacterium nucleatum. This is a bacterium that is normally present in the human oral cavity, and it's rarely found in the lower GI tract of healthy individuals, but has been found to be enriched in the colons of individuals with colorectal cancer. So we need to go back 12 years to that halcyon time of 2012. And this was when Fusobacterium nucleatum was the first bacterium to be associated with colorectal cancer through genome sequencing of tumors. We all take it for granted today that you can sequence anything fairly inexpensively and fairly rapidly. In 2012 was about the time that shotgun sequencing really came into its own, or if you will, online. And briefly, what folks did is they assembled bacterial DNA associated with the nucleic acid that they isolated from tumors, answering the age-old question, what was in the tumor microenvironment? The result inspired investigators to seek to define whether and how Fusobacterium nucleatum in the community of gut microbes could initiate or drive the development of colorectal cancer. So there's been a lot of work in this area for these past 12 years, with the key questions being which Fusobacterium species is most important to the biology of colorectal cancer, or is it a community of bacteria, including different members of this genus that can accelerate the formation of the cancer? And rather than summarize 12 years of literature, I'm just going to give you the bottom line up front from today's paper.
Before you do,
okay.
could I interject that one of the senior authors, Susan Bowman, was first author on a really exciting paper on this topic in Science in 2018, where they not only made the correlation between Fusobacterium and colorectal cancer, but they took a xenograft out of a human tumor, put it into mice, and then either treated or didn't treat with antibiotics, and found that when they treated with antibiotics, got rid of the Fusobacterium, the cancer actually got better. So that was the first indication that this was not just a correlation, but perhaps a causation. So really great motivation for her to establish her own group now at University of Washington, Hutch, and do this fantastic work you're going to tell us about.
I mean, this is, because it's open access and this is only a snippet, we're going to quickly summarize a lot of fine work. So I don't want to discount the amount of work that's in this paper. So I encourage you to go and take a look at it. It has something for everyone. But first, we need a few definitions to get us all on the same page. First up, what's a clade? In its simplest form, They are simply a group of organisms believed to have evolved from a common ancestor. So you then might ask, how do they differ from microbial strains, which again is evolution from the same species, but they're diverging? And this is a bit of taxonomic subtlety, but it's necessary to appreciate how these authors went about arriving at their title, because go back up to the top. It's all about this clade, and that's what's most remarkable. Microbial clades and microbial strains are simply ways that the taxonomists go to categorize microbes at a level below the species, but they represent different levels of genetic relatedness, and this is very important as we get to mechanism of how a microbe can literally transform our normal epithelial layer into an adenoma or the cancer. In the strictest sense, a microbial strain is a specific genetic variant within a species. Strains arise from mutations in the genetic code, or in the case of many bacteria, acquisition of new knowledge into that original or parent species, if you will. These mutations and additions can cause changes to the microbe's physical characteristics, like the additions of an antibiotic resistance trait, or its interaction with the environment, like the acquisition of a pilus that binds the attachment site more tightly, or like many of us are already familiar with, let's consider E. coli. There are many different strains of E. coli. Some we use in the lab that are considered very innocuous, like E. coli K12, while others, O157H7, can cause food poisoning and hemorrhagic diarrhea. Now, what's a clade? Well, a microbial clade is nothing more than a group of organisms that share a recent common ancestor. In the context of microbes, a clade can encompass multiple strains or even entire separate species. And clades are identified by analyzing the microbe's genetic similarities. And that's what these authors are going to do for us in this paper. So now back to the story. It's been observed in those individuals with colorectal cancer that they have high intertumor loads of one particular clade of Fusobacterium nucleatum over the one that is commonly found in our oral cavity. In fact, these high tumor loads are generally observed in concert. And Michelle already gave us the punchline associated with recurrence, metastasis, and poorer patient prognosis. And if you eliminate the bacterium, things can get better. So in this paper, the authors now are going to leverage a comprehensive collection of human colorectal cancer Fusobacterium nucleatum strains, carrying out extensive comparative genomic studies where they learned that there was a select clade within the Fusobacterium subspecies that simply predominates the colorectal cancer niche. And we're sort of setting you up for Dr. Martin in a few minutes, where he's going to be talking about microbial ecology and the wonders there. So really the two papers are complementary to one another because it's all about the niche. And in the elegant set of in vitro and in vivo functional studies that these authors are going to share with us, they were able to demonstrate, and I think equivocally, that the colorectal cancer clade was highly virulent. And here I believe their intent was to maybe argue was responsible for the disease observed and should become the primary focus for our subsequent mechanistic studies as to how the residence or the presence of this particular clade of Fusobacterium in our gut could be responsible for the pathogenicity. And pathogenicity is a fancy word that simply means generates the pathology of colorectal cancer. And so they're hoping to develop targeted inhibitors that we may be able to coat our gut with, just like the CDC recommends that we coat our guts with Pepto-Bismol or bismuth subsalicylate to prevent us from getting traveler's diarrhea when we go to different countries to worry about water. You know, it clogs up the receptors so the E. coli can't attach. And maybe we could do that with this. So this is really a neat set of data that they lay out for us. And it's a good paper to think about. The first series of experiments explores the distribution of Fusobacterium nucleatum within these two distinct niches, the oral cavity and the colon. So they first start by looking for niche-enriched genes and subspecies. I was pleased to see that they made a compelling argument for actually growing bacteria, these wee little beasties. And here they cultured 130 Fusobacteria, 65 were associated with colorectal cancer, and 59 were not. Now, appreciate that only last month I was offering to my poor dental students that Fusobacterium nucleatum is predominantly an oral pathobinot, which simply means it's a commensal until it's not. A good example that some of you might be familiar with is Helicobacter pylori. Half of planet Earth has this microbe colonizing their stomachs. Yet half the world doesn't have peptic ulcer disease or an adenocarcinoma of the stomach. So that's what a pathobinot is. It's a commensal until it's not. And so because Fusobacterium is normally in our oral cavities, and when we develop periodontal disease, we develop a condition known as acute necrotizing ulcerative gingivitis, which is a mixed infection of oral strep, oral treponemes, and you guessed it, Fusobacterium nucleatum, where the patient experiences a triad of symptoms: pain, ulceration, and these punched out interdental papillae. And if you look between your teeth, you see your gums, and the punched out means your gum is slowly receding. And this is gingival recession between your teeth and gingival bleeding with this real foul smell. But the smell can literally take paint from walls, or so I have been told by many, many people who actually suffer from this.
But worse than that is colorectal cancer.
Yes. So the authors include 81 strains of the oral pathbionot from individuals in the oral cavity who are not suffering from CRC. So what they first did is generate.
So wait, Michael, I had a question about this.
Sure.
So the smell, is it a diagnostic for having these? Is it sufficient?
Oh yeah, you can. When you get the odor of Fusobacterium nucleatum, you don't even have to go to the lab. You know what it is. It's quite
But
profound.
if it's populating your mouth, is it enough in there that people can smell it? Is it associated with like bad breath? Or?
Those short chain fatty acids are responsible for the halitosis, and you can achieve a critical threshold. Yes.
So
and
people who might even be able to guess that they have this.
Oh, yeah.
Before. Okay. Interesting.
And when it gets out of control, as your periodontal pockets increase in size as we age and our gums naturally recede, that's when the organism begins to take off, and it causes bone reabsorption and all sorts of other things. This microbe has a panoply of virulence factors that enables it to do things like cause colorectal cancer. So back to the story. These authors first generated complete enclosed genomes from the strains they cultured. And then they also did the corresponding epigenetic methylomes. Recall that when you methylate DNA, you can alter the expression from that nucleic acid by either turning the gene or operon on, or you lock it in the off state, or you lock it in the on state, depending upon what the methylation does to the cis-acting transcriptional element. In total, they had 146 unique Fusobacterium strains, of which 92% or 135 belong to Fusobacterium nucleatum, with 55 being associated with the colorectal cancer and 80 from the oral cavity. And if you look at figure 1A and B, you can see their scheme of how they went about doing this. And you may also notice it was assumed that the Fusobacterium nucleatum strains in the CRC tumors originated from the oral cavity. But Fusobacterium is rarely isolated from the lower GI tract, and they reasoned or hypothesized that the colorectal cancer-associated Fusobacterium strains harbored additional genetic repertoire or genetic extra stuff to facilitate their colonization in the human colon, and that concomitantly would then be responsible for the development of the cancer. Now, to formally test this, they examined these 135 genomes that they had sequenced using an omic visualization program called Onvio. And I dropped into the show notes a link to the Onvio website if you're curious about how this software package works. It's a nice way to visualize it. And it optimizes for assessing the pan-genome of microbes. And sorry, another genetic definition. Perhaps this snippet should be posted to This Week in Bioinformatics. Pan-genome analysis will identify all the genes in a species or their pan-genome, and then discerns between the genes conserved amongst most members of whatever you're looking at, whether it be a genus or a species or even a strain. And that's the core genome. And then the gene content shared among subsets of members or the accessory genomes. Not surprising, they observed that the accessory genome size increased as their number of Fusobacterium genome sequences increased, supported of the previous proposals that Fusobacterium nucleatum has an open pan genome. And that's really pretty spooky from the perspective of, are other microbes going to be causing cancers if they have an open pan genome? Which is, again, not all that surprising for organisms that can serve as pathbinons, where it's a commensal until it's not. Now, the authors offer that there were previously published studies using a statistically independent or this orthogonal approach, where they have seen the differential distribution of Fusobacterium nucleatum subspecies in tumor tissue from patients with CRC or colorectal cancer with mucosal biopsy specimens from patients with inflammatory bowel disease, where there were sufficient genetic differences between 4 Fusobacterium subspecies. And that's really the core of their paper. The 4 subspecies were Fusobacterium nucleatum anomalus, nucleatum, polymorphum, and not any relation to our host, subspecies Vincentiae. Vincent has his own Fusobacterium named after him. He also has a treponeme named after him.
Oh my gosh.
Yeah, there's Treponema vincentii and there's Fusobacterium nucleatum subspecies vincentii. Another inside bioinformatic tidbit, they also observed that the CRC-associated Fuso strains have a smaller accessory genome compared to the oral associated strains. And to me, that made perfect sense, given the type of mixed microbial infections that Fusobacterium causes in the oral cavity. And Fusobacterium nucleatum in the oral cavity is said to be a bridging species, where there's actually receptors on it that allow other microbes to attach to it because it's attaching to tissue, and together they cause this train wreck that is referred to as gingivitis or periodontitis that many of us go to the dentist for and get taken care of. So what they described is in this genetic analysis, the composition of the pan-genome by using this very cool gene clustering algorithm. And so they abbreviate that in their figures as GC, or gene clustering. And the prevalence data were used to define the core and all the other wonderful aspects. But the next bit is the big deal, in that from their genetic analysis, they found the proportion of subspecies by niche. So this is the location these microbes are principally found. Of the 4 subspecies, only Fusobacterium nucleatum animalis was significantly associated with the colorectal cancer niche. That's the big headline news. They found one substrain that was associated with the colorectal cancer niche, which
and they could then look at the genome and say, aha, these are the genes that are distinct. So let's get into that. What do those genes confer? Why are they associated with tumors?
It's because of the metabolism. And it's principally they identified these 241 clusters and a group of genes, mainly groups of metabolic genes. And they compared and contrasted what was different between those microbes in the oral cavity. These are the same nucleatum pangenome, if you will, and those that they were associated in the cancer cohort. And then they started to search for unique virulence factors, and their analysis revealed the possibility that this one strain, Anomalous, that was associated with colorectal cancer was further subdivided into Anomalous clade 1 and Anomalous clade 2. And then using principal component analysis, and this is beautifully illustrated in figure 2, they showed that Fusobacterium nucleatum Anomalous clade 2 is associated with human colorectal cancer in that it has a larger chromosome than do the Fusobacterium nucleatum clade 1 strains, as well as a different or distinct genetic composition. And that's why I brought up the E. coli K12 versus E. coli 0157 earlier. You can still be named the same, but you can behave very differently. And just for good measure, they had the methylation pattern data, and they learned that FNA clade 1 and FNA clade 2 have distinct methylation patterns in their sequences, specifically in the 16S ribosomal RNA gene, which will probably serve to ring the bells in product development labs and diagnostic companies. For if you can tell the clades apart and add that to existing screen tests, you may help in future diagnostic studies. And we've all seen the ads on the evening news for Cologuard. And Cologuard is a simple test that you can do at home and you mail it back to them. And what Cologuard does is it's a multiplex diagnostic screen that looks for abnormal DNA methylation in 2 specific genes, NDRG4 and BMP3. And it also screens for 7 specific KRAS gene mutations, as well as looking at the beta-actin gene and looking for fecal hemoglobin. Just imagine if we could add this clade to a Cologuard-like test, could it be more sensitive and pick up that cancer sooner? But back to the story that they're telling us. I'll not go down my blind alleys. So what's unique about clade 2, Anomalous? Well, they offered it has this gene, FAP2, which has been previously implicated in promoting cancer. They also commented that clade 2 might be better suited to survive its trip to the acid of our stomach.
I love that data. That was really an exciting part of the paper.
It's so cool. And FNA2 has also nutrient scavenging mechanisms and is more metabolically active than FNA clade 1. Now, collectively, all of these data support the idea or notion that FNL clade 2 has distinct capabilities compared to Fusobacterium nucleatum anomalous clade 1. So what's next? Similar to fulfilling Koch's postulates for Helicobacter that many of us teach our undergraduates about, that Helicobacter can cause both ulcers and stomach cancers. So they did a pseudo Koch's postulates fulfillment experiment. They asked the question for the ability of Fusobacterium nucleatum C2, its impact in intestinal tumorigenesis and metabolism. And this is beautifully described in their fourth figure.
In a mouse model, not humans.
It's a mouse model, but we get to humans too. So what they used is 6 to 8 week old APC min plus minus mice. And the model requires you feed them streptomycin and dextran sulfate to alter the native microbiome and induce colitis. So the mice then are orally gavaged with a vehicle control, that's arm 1, or mix of FNA-C1, which is arm 2, or FNA-C2, which is arm 3. The mice are then monitored until the endpoint at 6 weeks post-gavage when they reach 15 to 17 weeks of age. In the second panel, they answer the question, did they get cancer, by simply counting the number of adenomas that were detected. In the control, vehicle only, no cancer. In FNA clade 1, one animal had no adenomas, and so there's 8 animals in total. 5 had 1 adenoma, and 2 animals had 2 adenomas. And 1 had 3 adenomas. But in the clade 2 arm, all the animals had at least 1 adenoma. 5 had 3 or more adenomas. 1 had 6, and 1 had 8 adenomas. So this actually proved that clade 2 has something extra special that enables it to form these cancers in this animal. So
and importantly, it's not just that these can just latch onto cancer cells and grow there, but rather they're triggering something, potentially interfering with, triggering inflammation through nitric oxide, one of their metabolic experiments they did.
Yeah. And so the last experiment, this is in figure 5, is, we've all heard the story that, mice are not people. So here in figure 5.
Yeah, that's some story, isn't it?
What?
Well, what is it? Vincent, monkeys might.
No, wait. Let me tell you what it is. It's a criticism of animal models, which are not faithful to humans. Mice lie, monkeys exaggerate, and ferrets are not humans.
That's it.
Okay.
But this data is really compelling.
It is. And so what
don't be too compelled, though.
Well,
how compelled should we be? 50% compelled? 20%?
Let's see. Tell us,
Take us home, Michael.
All right. So what they did is they analyzed stool metagenomic data sets from publicly available cohorts of patients with colorectal cancer. So we've expanded our N up to 627. And they also looked at healthy individuals that do not yet have a diagnosis of colorectal cancer. So here the N is 619. FNA, so the Fusobacterium nucleatum anomalous, was detected in 29.2% of the stool samples from patients with colorectal cancer, and only 4.8% of the stool samples from healthy individuals. Then using, and here we have to use some fancy statistics, using a metagenomic analysis of standard mean differences using a random effects model, they found that both clades, FNL clade 1 and 2, have a significant pool size effect, but the effect size for clade 2, the one that matters from the animal model, was larger than that for FNA clade 1. Notably, in the absence of FNL clade 2 co-occurrence, and clade 1 and clade 2 can coexist in some of these metagenomic stool samplings. if it doesn't exist, it was not, FNL clade 1 was not significantly associated with the colorectal cancer cohort. So this is nicely summed up in the graphic 5D, where their data showed that FNL-AC2 is more prevalent and abundant in the stool of patients with colorectal cancer than FNL clade 1. Now, these culture-independent human specimen analysis support strain-level genomic diversity that clade 2 is the dominant CRC-associated clade from their principal component analysis, while at the same time highlighting the significance of the in vitro data, which we haven't even touched on figure 3, and the in vivo data, which we just talked about in the mice, is in figure 4, and the increased virulence and tumor genetic potential for clade 2 of this nucleatum anomalous over nucleatum anomalous clade 1. And so, they have really given us a complete story from which we can develop mechanistic studies going forward. What's next? I would expect we're going to begin to see knockout experiments out of the FNL clade 2 group of strains. First, a unique colonizer of the tumor microenvironment, and then using that family of knockouts, it should then reveal which genes or metabolic pathways or receptors or pili are involved in the biofilming, loving, strict anaerobe that's known for its ability to form these mixed microbial communities that can then generate substantial community-altering phenotypes and this pathology. As remember, this is the beast that's implicated in this acute necrotizing ulcerative gingivitis, which is pretty profound, and how such a beast has gotten a leg up in its ability to initiate or promote human colorectal cancer is now been placed front and center for investigators across the world, thanks to these authors. It's really a great study to look at.
Yeah, and also motivates some diagnostic tests.
Yes.
I would just caution that it's never so simple.
Yes.
Right? Not all colorectal
cancer is usually takes 12 different mutations to get. You need a driver to get the cells to divide forever, and then a bunch of other mutations come in. So maybe you get rid of the bacteria, it's maybe not going to do anything, or maybe reduce it. But so I would be
or take longer,
cautiously
more years.
Optimistic, right? And every cancer is different slightly, so it's
right.
nice. But
I had a question about this one, though. It's interesting in that it's just this very small group, so it suggests kind of very direct inflammatory response is what I was interpreting it as. But there's so many things that, and then there's also inflammatory bowel disease. And I don't know if those things lead to cancer, or does this suggest a more specific inflammatory route? Not knowing anything about inflammation other than sort of generally, like, is there a possibility that some cells are giving an inflammatory response to these bacteria specifically, as opposed to a general inflammatory response? And that's why.
Petra, I often get students who'll ask me, is the answer A or B? And then I'll say yes.
Exactly.
And I think there's some truth to that here. And I wanted to also point out some of the news and views material. Actually, I'm not going to say they dampened the positivity that Michael is showing, but they did make some cautionary statements there. And I think, I view data as my enemy. It's always trying to fool me.
Good point.
Right? Yeah.
Absolutely.
I had a question for you, if I could, Michael.
Sure.
Are these Fusobacteria, are they invasive on the tumor? Because I have thoughts about this.
That I don't know, because they didn't comment. They didn't discuss it, and they effectively isolate the tumor, and they grind up the tumor, and they isolate the nucleic acid. Mm-hmm. That's the level and extent. How tightly they're affiliated with the tumor, I don't know. And whether or not they're internal to the tumor, as Vincent says, it's complicated.
But in earlier work, they did show that the Fusobacterium stayed associated even when the tumor was metastatic.
Right.
So it is
and that's what FAP2 does. Yeah, I got it. And this reminds me of the old school work where Well, first off, because tumors have not been properly vascularized, you have all this like heterogeneity within the actual mass, and those provide different niches. And I know that you can get, for example, salmonella colonizing anoxic regions of a tumor. That's something that some folks have tried to work on. And that's what I was thinking with this a little bit, because when you're in the gut, you're surrounded by like 10 to the 12th microbes per cubic centimeter, right?
Yeah. It's really a conundrum. And Petra sent me a question yesterday wondering why we're beginning to see the age that we detect colorectal cancer dropping and what's going on. And so I asked our cancer folks, and they came up with the usual suspects: unhealthy habits like obesity, physical inactivity, smoking, and potentially vaping. Vaping may be in there, and a diet low in fiber and high in processed meats and red meat. They're bringing out the old tropes as to what causes cancer. But I think it may be something even more subtle, because this generation of 20-somethings has seen a greater use of antibiotics than we did growing up. Mm-hmm. I mean, I can count on my right hand the number of times I had antibiotics before I was 25. So I'll drop into the show notes two things one from MD Anderson on what young adults need to know about colorectal cancer, and what millennials and Gen Zers need to know from Yale Medicine.
And those of us over the age of 50 as well.
Yes.
Exactly.
Thank you, Michael. All right. Mark, do you have something for us?
I do, and I can keep it relatively short because I know everybody has an issue of time, but I just wanted to tell Michael, speaking of antibiotics, I was treated for acne with about a gram of tetracycline a day for 7 years.
Wow.
So when I took the microbial diversity course, we were with Abigail Salyers. I took it, and we isolated some Bacteroides. I did from me, and like 30% were still TET resistant. I just thought I'd share that.
So
there's changes.
Is that different though? Like if you compare you to somebody who didn't receive that? Because the question is, how long is that going to last? Because that TET resistance should be essentially a negative selection on them when you remove the tetracycline.
The plural of anecdote is not data, Petra.
Yes.
So I want to be right.
This is a real anecdote. Okay.
There was a young woman who was from Belgium where you can buy antibiotics over the counter, apparently, without prescription. And hers were 80% TET resistant. I just wanted to tell you that. Isn't that amazing?
Yeah, I don't think we use TET anymore. Is that correct?
I do in the lab.
Yeah, no, in the lab, right. I think they stopped using it.
It's gone to doxycycline.
Yeah, doxy.
Right. Well, hey, friends, I am happy to tell you about one of my favorite topics, and it all derives from the business of our focus on single colonies, pure cultures, that kind of thing. It's not how real life works. For example, when folks first started studying biofilms, they would, look at one microbial species. I used to look at Pseudomonas putida forming biofilms. Not a problem, but that's not how biofilms normally form. So one of the problems that we have is a conceptual change in microbiology about the interactivity of what's actually going on. And one of the greatest examples of how to study it is looking at a three-membered grouping. And this comes out of the wonderful work from Joe Handelsman's group at the University of Wisconsin in Madison. And this is actually from MBio. And oh my God, I was going to write this down, but I'm not sure I got the date. Oh yes, it's March 2024. There we go. So we're up to date. I know because Michelle would just look at me if I didn't say it. So in any event, what I was going to say is the title of this article is Surface Colonization by Flavobacterium johnsoniae Promotes Its Survival in a Model Microbial Community. And I have been fortunate enough to meet a number of members of this crew, so it was a lot of fun. Magesh, Hurley, the inimitable Julia Nepper, Mark Chivet, Jonathan Chope, Chao Li, David Beebe, and of course, Joe Handelsman. And several years ago, this group discovered something very interesting. And I think I've talked about it before, but I just want to bring everyone up to date. We're used to the idea of single colony isolations. But what if you streaked out a bacterium several times to single colonies, and then you saw things sneaking out from that colony later? It really makes you think about colonies. And this is the case with a lot of natural isolates of Bacillus cereus. Now, Bacillus cereus, as a gram-positive, has about 50 cell layers thick of peptidoglycan. And when this was first observed many years ago, a lot of people started thinking, what if there are microbes that hang out and use that huge amount of peptidoglycan? And in fact, they do. I tended to think of them as peptidoglycan surfers because that always made me laugh. And they found the Flavobacteria were doing this. But the group decided that the best name for this was Thor, the hitchhikers of the rhizosphere.
I love that.
So, yes, it's wonderful. And in order to study this, they found out a couple of things. It's this update that I find interesting. And I just wanted to give you that background because the focus here is on Flavobacterium. But remember, you have Bacillus cereus, Flavobacterium johnsoniae, and Pseudomonas corinensis. And they actually are all interactive. And some may look down their nose at only having 3 members, but the complexity among these 3 are quite remarkable. In addition, there's always a problem when you look at a non-canonical strain to work with. I've spent my life working with undomesticated microbes, which are just heartbreaking because things that work very easily in one strain don't work well in another. But the good news is all 3 of these members can be worked with genetically, and Mark McBride's work with Flavobacterium was really applicable here. So the simple hypothesis here is as follows, that surface colonization by Flavobacterium is necessary for success in the Thor system. That's exactly where we're going to start. Now, I could spend time talking about the Thor interactions, the fact that the Pseudomonas is making an antimicrobial and the Bacillus is turning that down and the Flavobacterium is taking advantage of both. You get the idea. So the focus here is on Flavobacterium. And they did this something that I know I should have heard, but I hadn't heard of it before, insertion sequencing, in-seq. I'm used to the idea when you do transposon mutagenesis of making 3,000 or 4,000 mutants. And in fact, with in-seq, and I keep wanting to say in sync, the boy bands, that's my fault, not yours. 75,000 mutants. And what they were able to do which I think is remarkable,
it's impressive,
is they made a challenge. And what they did is they asked what adhered, for example, to sand. And so they were able to look at the planktonic survivors, as it were, of this process, and ones that had adhered to the sand grains. And what they found is about 25 different mutants that appeared to be important for sand colonization. 21 of them seemed to be involved in reducing colonization and 4 with increasing it. What does that mean? After they do this experiment, they're able to ask the question which of these mutants were more prevalent or less prevalent after passaging them on sand. I hope I put that well enough.
Mm-hmm.
And 40% of these bad boys, I shouldn't genderize them, but you know what I mean. 40% of these mutants were in unknown. And I love me some unknown hypothetical genes because that's exciting to me. The rest, and I'm not speaking down about them at all, were involved in cell wall membrane synthesis or transport or the synthesis of metabolites.
Yeah. And Mark, the authors make that point too, since most of the literature is culture, pure cultures of one bug. These are important for living in a community, so it makes sense that they haven't been uncovered.
Well, no, but I'm just saying that because when I find something in my own modest work where the gene that we get is unknown, that excites me.
Yeah, it's pleasing, and it motivates them to do these more complex multi-species colonies.
Michelle, the look on one of my undergrad's face when she said to me, "Nobody knows what this gene, if it is a gene, what it does." And And I said, and this is relevant because you have to find out if it's really due to the mutation. So, as I said, they decided to kind of focus on 9 of these genes. And I believe, unless I'm mistaken, that's table 1. And they did in-frame deletion. And it's kind of hard to do this it's easy to do it with E. coli. Yes, I hear it all the time. And Salmonella is actually E. coli. I hear that all the time. But in fact, Flavobacterium is not easy. And again, Mark McBride had spent a lot of time and effort creating genetic tools for the study of the Flavobacteria. And it appears to be these 9 genes they're focusing on. They made in-frame deletions of each of them to see whether or not they had an effect on what took place. None of them, when they made these in-frame mutations, had any effect on planktonic life, which makes sense. But they had a significant impact on colonization, and that shouldn't be a surprise because that's what they were looking for. So then they introduced these wild-type copies of the genes on plasmids, and they showed that correct the mutant phenotype. It's just a beautiful, clear explanation. I don't know off the top of my head, and this is on me, if there's a copy number issue with the plasmids that they're putting back in Flavobacterium, because that has confounded me in the past. But what's interesting is it doesn't look as if the defects are in attachment per se, but they do seem to affect proliferation. And if you look at, and I hate to be so generic, biofilms in general, there's an attachment step and then proliferation on the surface. They went on to show that they saw the same mutant phenotype on glass beads and polystyrene. This is fabulous. Mm-hmm. Now, Flavobacteria generally use a type 9 secretory system as part of their gliding motility Swiss Army knife. And they were able to show that these mutants seem to impair sand colonization in the presence of root exudate. When we talk about the Thor system, remember that this is essentially part of the interactivity with soybean roots. So that becomes important. Michelle, they went on and did biofilm formation the way that I teach undergrads with crystal violet, and it was impacted there. But friends, I know you want to hear about Zorbs.
I love their terms.
Yeah. And,
Thor,
and Vincent, do you know what a Zorb is?
I don't. Tell me.
So the way that I heard it is there are these giant transparent balls that you get into and you run downhill when you're in New Zealand or places like that. So in a similar way, what they are little motile biofilms. So in other words, there's some cells at the base and they're using gliding motility, and there are a bunch of, and I don't want to say hitchhiker on the hitchhiker, but you get the idea. There's very specialized equipment. And when you look at some of the images, and oh my goodness, what figure is that? Let me get there. You can light up. Thank you. I talk to my computer. Don't you, by the way?
Of course.
This is figure 5.
Thank you so much. I just got there.
Yeah.
And you can see, and may I tell you, and I don't want to brag, I've actually seen these for real, and it's wonderful. And like these little tiny, for lack of a better term, little spheres moving, and they move, and there are movies associated with this as well. Yeah. And so you're able to see that these motile biofilms are also being impacted by these types of mutants. So what I like is that when you look at table 1, it shows you the 9 types of genes that seem to have an effect, and it gives their annotations. And I draw your attention to table 2. And in table 2, 8 of the 9 seem to have pretty much, well, maybe 7 of the 9 don't really get much going on, but you can see that a couple of them are different. So we've talked about sand colonization. We've talked about biofilms. We've talked about Zorbs. Now, one more thing to tell you about is whether or not it causes colony expansion. And I've seen this when I'm teaching the Tiny Earth program here at the University of Puget Sound. When we're testing for antibiosis, we'll spread on the surface of a plate a particular organism, and then we'll stab in a challenger. And sometimes you get very unusual effects where the bacteria that you stabbed into the background will really expand like crazy, looking like fingers spreading out, and other times not. Well, this is exactly what they're talking about, because with the Thor system, Bacillus cereus, and there's a lovely photograph of this in one of the figures, you get all this expansion. So clearly they're interacting in a way. And as you can see, for 1, 2, 3, 4, 5, 6 of the mutants, you have the wild type level of colony expansion, but then you get more for some of them. And that moves us to the final two categories. I am trying to go quickly, friends. Community planktonic and community sand. And the whole thing to tell you about here is there doesn't appear to be a major effect for 8 of the 9 mutants for planktonic effects. And in the community on sand, all of the mutants seem to have an effect. Now, what does this really mean? I've rushed through a lot of stuff, but I think it's important to find out that what we're doing is dissecting sociomicrobiology among the members of the Thor community. So we can look at it in terms of fitness. We can look at it in terms of the spreading colonies. We can look at attachment. We can look at whether or not, I mentioned fitness already, but you get the point. This is such a ripe system to really bring new tools to. And I cannot wait to apply what's being learned here to other systems, perhaps one associated with animals. And that's kind of what I wanted to say about it in the interests of time. What can I answer or pontificate about with all of you?
Well, I'll just add that they also point out that this consortium is likely important for agriculture as well. So it gives us inroads into that, which of course is hugely important for feeding the population.
And Michelle, I believe that this particular strain of Bacillus cereus, UW, is the one that makes a fungicide.
Interesting.
Yeah.
Yeah, I think you're right. I think you're right, Mark.
Yeah. Yeah. Eric Stab was the one who actually has a patent on that. So pretty cool work.
So this the lack of effect of deleting these genes on planktonic growth is interesting. It reminds me of a virus story, because if people were still studying growth in broth and didn't know anything about biofilms, you would delete these genes and say, they're non-essential.
Yep.
They don't do anything.
And therefore they're unknown in the
are useless.
Useless. For
really
many
like
viruses, we grow them in cells in culture in the lab. We delete genes. There's no effect. We say, they're not essential. I remember going to Columbia, my chairman, Harry Ginsberg, used to rail against that. He said, there are no non-essential genes. They have to do something. We just don't know what they do.
True.
And it turns out if you put those viruses in animals, then those deletions have an effect because they're immune antagonists, and you just don't have immunity in cell culture. Yeah. So it's a theme, right, in microbiology.
Wow.
And this comes back to context.
Of course.
And so even within the 3 members of Thor, I mean, I want to know, please forgive me, I've been after this lab for years. I want to know if the Flavobacteria are drilling down and that we can see them inside the peptidoglycan, or are they just sitting on top? Because they definitely are able to feed on the peptidoglycan fragments that Bacillus cereus makes.
Or are they stealing?
Really interesting.
Are they stealing the disaccharide as it gets sent to the top to expand, and they drill down and they grab the disaccharide as it comes across the membrane? So they're literally getting spoon-fed by the microbe.
This is knowable. All of this is knowable.
Right.
You can easily,
knowable.
Technically it's possible to look at the single cells by confocal and see their interactions in that. People have done that inside mice, so it should be relatively straightforward. But it would also be really cool to get some transmission EM or scanning. Transmission probably is what's necessary here to see, but you could also label the peptidoglycan and see where it's going.
And there have been some papers on this, Petra, which are just wonderful, where I didn't know you could put peptidoglycan in the media. And And then you get like zones of clearing, even on that level, perfect for students.
But I would be really curious, is Sirius actually shedding its peptidic glycan at a high rate? I mean, what happens if you treat them or you have mutations where they do shed it more, where there's less cross-linking?
Now, this is why I'm glad you're here.
Lysis, I mean, I'm just like thinking of all these things, and these Y genes are really, these unknowns, Y genes in E. coli, but other ones are really interesting.
I'm really happy to hear that, Petra, because I'm so used to the idea of a lot of bacteria shedding bits of their peptidoglycan as MAMPs.
Right.
And I'm so used to that idea that it doesn't occur to me, has it been measured? Right?
Yeah, I mean, we talk about it and we talk about bacterial warfare, right? There are definitely, bacteria that secrete things that then cause shedding, right? That might interfere with some of the activity. There's some really nice work in Acinetobacter from the Feldman lab here. But, this is really interesting. Like, what is the warfare at the single cell level? And I think that's kind of the next step in one way is to really think about it from a molecular perspective. And then this perspective about close association with the plants is really interesting because can you, not only for the fungicides, but maybe helping them get nitrogen and other things. That's sort of an agricultural perspective. There's so many different levels here. It's very cool.
And I love telling students about how primary it is to actually look at your plates and thinking about your plates. Because if I were streaking up something a couple of times and then I shoved them in the cold room and came back a week later and I saw little fingery flavos coming off from it, I would have just thrown everything away.
Like they're contaminants, right?
Yep.
But Mark, your point about looking at the bacteria brings me to the first author here, who is a fifth-year graduate student in the Handelsman lab named Srutha Magesh. And she is from India. She's a first-generation PhD student. And as a kid, she came across a show called FAQ on the Pogo TV channel. And it just showed science behind a lot of just very common things that we observe. And she said that really got her excited in biology, and she was fascinated by it in high school. She also began doing science communication through middle school. And then as an undergraduate, she was able to work on two different research projects. One focused on genetically engineered bacteria for remediating mercury-contaminated sites, and another on harnessing enzyme and biosurfactants from microbial consortia to remediate oil sludge waste. So you can see this set her up perfectly for the work that she's doing as a graduate student. But she credits an amazing program that is a collaboration between India and US. The fellowship is called the Corona program, and it gave her the opportunity to visit the US and work in the lab. And she actually was in Joe Handelsman's lab as a visiting undergraduate, and that was her first exposure to Thor. So she's forever grateful for that opportunity as an undergraduate, and kind of opened her eyes to pursuing science as a career. So she has advice for aspiring scientists and fellow students. She says science is a roller coaster of excitement, difficulty, and occasional setbacks. Embrace the curiosity, roll with the failures, they'll happen, and never underestimate the power of teamwork. Trust me, she says, in the end, it's a risk worth taking. The satisfaction of making a meaningful contribution makes every hurdle worth it, so never give up, and don't forget to take breaks and enjoy the journey along the way. And what she enjoys when she's taking a break is traveling, spending time outdoors. Also, she's recently taken up skiing, and now it's her favorite winter pastime. So again, she's incredibly grateful for that fellowship that allowed her to come and experience Joe Handelsman Lab and be introduced to Thor as an undergraduate, and it set her on the journey, and including leading this beautiful paper.
And she's a wonderful person in person. That's
Hi.
Too many impersons, but you know what I mean.
You know her?
I met her when I was visiting one time.
Great.
I've met several of them, and they're just wonderful people.
I'll bet the the excitement that the two of you shared must have taken it up to 11, huh?
That's with Dr. Nepper. That's how I was. But I think Shrithi was a little, I'm a lot. Let's just put it that way. I am an acquired taste, small batch, artisanal.
Thank you, Mark.
You're very welcome. I really enjoyed talking about it. I hope everyone got something out of it. You never can tell. And this is interesting. You think, and I get this a lot from students, what can I learn from, for example, a plant system like this? Well, a great deal.
Mm-hmm.
And it's encouraging to see that we're able to do more mechanistic science with consortia and not just a pure culture in broth.
Sure.
Yeah.
I think we've talked about Thor before on TWiM, right?
That's correct.
Yeah. It rings a bell.
I dropped the Thor paper into the show notes. So if our audience wants to look, it'll be in the show notes for you.
I think it's also might be nice to drop a little something about Gobind Khurana, who's helped figure out the genetic code, whose fellowship supported the first author of this paper.
Oh, yes.
Because he's very interesting.
I remember when I visited Wisconsin, they showed
he was at Wisconsin, right?
They looked out the window and said, that bridge they built for him so he could walk to his lab.
Wow.
Yeah, he's definitely one of the most important faculty, famous faculty from there.
All right, that is TWiM 308. You can find the show notes at microbe.tv/TWiM. If you enjoy these conversations, we'd love to have your financial support. It would be U.S. federal tax deductible, as Microbe TV is a 501(c) nonprofit. You can go to microbe.tv/contribute for more information. And if you have questions or comments, twim@microbe.tv. Our guest today from the University of Puget Sound, Doc Martin. Thank you, Mark.
You're very welcome.
And good luck with Matters Microbial. Keep it going.
I'm loving it. I hope I get many more listeners and viewers.
I think we're going to say we're Matters Microbial adjacent.
Yes.
That's right.
Yes, we are Matters Microbial adjacent.
I did link to Matters Microbial in the show notes. So
we will.
Well, that's nice.
You will be more adjacent once you get on his program.
Yes.
That's the requirement to be adjacent, right? You have to come on.
You get a gift package when you come on my program.
Pin?
Gift package.
Custom pins? All right.
You do.
All right. Michelle Swanson is at the University of Michigan. Thank you, Michelle.
Thank you.
Michael Schmidt, Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levin, Washington University in St. Louis. Thank you, Petra. And I am Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.